We're going to talk now more about properties of enzymes. That's really the thing that we're focused on in this metabolism course. Rather than the cell biology course, I try to focus more on some other types of structural proteins. This is really talking about enzymes. I'm not going to spend much time at all. I'm going to have these are going to be on your PDF files of your PowerPoints. I'm going to give to you guys to look at if you need to write notes on, but I'm not going to really talk about primary, secondary, tertiary, or quaternary structure because you should be pretty familiar and understanding of this already. This is a question you should be able to answer. If you do not, then I would review the previous lecture slides and then move on to this lecture. We're going to really talk about some of the things that make enzymes unique. One of the things that they need are a lot of times there are different things that are going to be allowing the enzyme to interact with the metabolite or the ligand. So these are two different terms that are often used for the same thing, really, right? So something's going to be the reactant, something's going to be the product. That's something we refer to as a ligand or a metabolite. That enzyme is sometimes undergoing a chemical reaction, but it's always recycled back into its original form, and hence it's called an enzyme or a catalyst, right? Enzymes are essentially catalysts, but it's in the biochemical world. You should be able to list and know all of the intermolecular forces. If I show you a ligand and I show you an enzyme, you should be able, and if I say, hey, look, there's a binding pocket, what kind of intermolecular forces do you see? Do you see hydrogen bonding? Do you see ionic bonding or electrostatic bonding? Van der Waals, dipole interactions. So what are you seeing there? Can you recognize it? If you can't, go back to the previous lectures, watch those again, understand and know that. Here I just want to step back and make sure that we have the terminology we need to move forward together in this discussion on catalysis and enzymes. There are different ways of showing this that you can see that previous slide. I have an image of a cartoon, right? And this is the green is the enzyme, and then we have the ligand in the middle. This is a three-dimensional rendering of a protein structure. So this is actually data that has been fed into a computer program, and you can see this little stringy stuff is the backbone of the peptide backbone, and then we have alpha helices and some beta sheets you can kind of see around here. And we also have within that is the substrate or that component that the enzyme is going to bind. And this is also labeling the active site. And this is where that enzyme is going to bind the substrate and create some type of chemical change in that reaction. So just so you can recognize, sometimes some of this stuff is just kind of going to be cartoonish. Sometimes it will be actually from data that was developed when to create these three-dimensional structures of a protein. We can also look at this. This is another image, just different rendering of that data that has been generated, usually through X-ray crystallography or NMR, in order to know where those the atoms are within that molecule. And here this is just a different structure, a different way of looking at it, where instead of the strings of the individual backbone, you just kind of get this globular protein three-dimensional structure of this is what the things are, but we don't really see the individual alpha helices or beta sheets. It's just showing you the different amino acids that might interact with the substrate and where the substrate would be found within that binding pocket. So these are different ways that we will show you how these proteins, enzymes look. And sometimes they'll be simplified into something like this so that you can look more closely at the enzymatic binding pocket where these chemical reactions are occurring. Here are just some of these types of intermolecular forces, just to reiterate, right? Serine, because it has this alcohol group, it has a hydrogen that's attached to an oxygen, it can hydrogen bond. Phosphate group, right? It can't hydrogen bond, but it can be involved in hydrogen bondings. So how you say these things are important. It can be confusing, and I know if you've heard this from other faculty members where, well, a phosphate group can hydrogen bond. Just note, for me in my classes, double bonded oxygen cannot itself hydrogen bond. It's really phosphate groups can dipole bond because it has that electronegativity increase on the oxygen compared to the phosphate group. Here's an electrostatic interaction, right? If we have an arginine side chain and we have an amine functional group that has a positive charge, and let's say we have a phosphate group that lost an alcohol or a hydroxyl group here and is negatively charged, right? When full positive and full negative charges are interacting, that's when we can say it's an ionic bond or an electrostatic interaction. Here's some more. Here's another hydrogen bond. Nitrogen is more electronegative than hydrogen. So this is hydrogen bonding here. This is interacting as a dipole because we have negative charge here and it's interacting with that hydrogen. This hydrogen is hydrogen bonding with this carboxylic acid. Right, so all these different hydrogen bonds are shown here, dipole interactions as well. So it's really important that these side chains are really helping to modify and influence the structure. They come together, they make these types of interactions, not only with different ligands, for example, for AMP, cyclic AMP that's shown right here, right? Not only for different ligands or metabolites, but also just when these things are being folded, right? We talked about the primary, secondary, tertiary, quaternary structure so many times. What is holding that together are all of these different, both the side chains as well as the backbone interactions. Here's a slide just to kind of go over, just to make sure that you know the verbiage that's being used in biochemistry. So enzyme, again, is a catalyst, but it's a protein catalyst. An active site that's where we are going to bind in the enzyme pocket. So we call it an active site because that's where the activity happens. And a substrate, again, I mentioned, can be substrate, ligand, metabolite, right? Multiple words that really mean the same thing, but I just wanted to cover that so we're all on the same page here moving forward. This is an example, just kind of a cartoon of maltase, right? The enzyme name that is breaking down maltose. This is a carbohydrate, maltose. Notice the OSE ending. That tells us it's a sugar. The ASE ending tells us that it's an enzyme, ACE, right? If we say protease, we're saying it breaks down proteins. A ligase, right? We're ligating things together. We're gluing things together. Maltase, right, is breaking down maltose. So these are enzymes. Maltase is perfectly shaped, right? That active site's perfectly shaped to accept a maltose molecule. It can break 1,000 maltose bonds per second. And it's only going to bind to maltose molecules because of that specific shape of the active site. There were 5 different types of reactions that we talked about for organic chemistry. There are 6 different enzymes that are classified into 6 classifications for metabolic enzyme-based reaction. We have the oxidoreductase, isomerase, transferase, hydrolase, ligase, and lyase. So these 6 you need to know. They're similar to the organic chemistry versions that we have previously discussed. When we're talking about these different types of enzymes, sometimes some of these names will be involved in them, and sometimes they won't be. But a lot of modern systematic naming of enzymes generally has the substrate, identifies the substrate first, and then the second part is the enzyme subclass names. For example, pyruvate carboxylase, it's going to carboxylate a pyruvate molecule. It's difficult sometimes because depending on how you're looking at the reaction, most organic chemistry reactions are reversible, and there's a weird reversible symbol, but they can go back and forth. So sometimes it's confusing, right? The pyruvate carboxylase may catalyze both the forward and reverse reaction, where pyruvate then, if it's the reverse reactions, technically, right, it's not the substrate, it's actually the product. I'm going to walk through these different types and give you examples of them so that you have an idea of what is happening, what the chemical reaction is that these enzymes are catalyzing. So here I'm going to start with the hydrolase, and This should look somewhat familiar to you, right? We've previously been talking about amino acids and peptide synthesis. This is actually what happens when we break down peptides. So if we have a protease, right, something that's going to break down a protein, it is going to recognize this amide functional group on the polypeptide. And a lot of times different proteases are actually going to recognize either downstream or upstream certain and specific side chain moieties. So these side chains, right, if there's a serine or a tryptophan or a tyrosine, right, it's going to recognize, the enzyme's going to recognize this as part of the binding pocket. And the active site's actually going to work on this amide functional group. We're going to use water. Remember, when we took amino acids and glued them together, we had a water molecule left behind. So we took 2 amino acids, we put them together, and water was formed and a peptide. So the reverse reaction actually uses water to break apart this amide functional group. And what we're going to do is we're going to, remember, make a carboxylic acid here. So if we rewrite water, sometimes it's nice to write water like this. So what we're going to do is we're going to take this hydroxyl group and attach it to the carbon. And we're going to take this hydrogen group and we're going to attach it to the nitrogen. And that's essentially what's going to happen here. You'll see that we have this CO and we have an NH3+. So obviously this oxygen, because of the pH usually, right, we can have that hydrogen. And if this is neutrally charged, we know that this likes to get a positive charge. So they're just showing you kind of what's going to eventually happen. But if we follow along the organic chemistry pathway, right, this hydroxyl group is going to attach first, and the electrons are going to flow from this bond over to the nitrogen. That's going to be then have a lot of negative charge on it. It's going to be attracted to this hydrogen. And then that's still, right, this, because of the pH of the system, generally, we're going to end up with a negatively charged carboxylic acid. It doesn't have to be a peptide, right? It can be anything, something that's going to eventually split apart. So this is similar to a substitution reaction. You recognize this type of reaction from our organic chemistry types of reactions. This is called hydrolysis because we're using water to break apart these bonds. A ligase is the next type of enzyme, and this is where we are gluing things together. Similar to an addition reaction. We're starting with 2 reactants here, carbon dioxide and pyruvate, and we're going to get a bigger, longer chain of carbon, oxaloacetate in this instance. ATP is not really involved, at least in terms of this specific reaction. It's used as an energy source. So we use this as energy it breaks down to ADP, inorganic phosphate. So this is just to get this reaction to work, and we'll talk about that later. But the reaction that's happening here is this gluing together of these 2 compounds to get a longer one. So ligase glues things together. We can think of this if we're trying to relate it to those organic chemistry terms. It's similar to an addition reaction. Isomerase reactions, similar to a rearrangement, right? Because we're making isomers. So triose phosphate isomerase is an example where we have a ketone here and an alcohol. We're going to switch it. We're going to switch that carbonyl group to the outside. It's not a ketone anymore, but it's still a carbonyl group. Right now, this is an aldehyde, and that hydroxyl group goes into the middle carbon. The next reaction that I'm going to talk about is the oxidoreductase. So this is going to oxidize or reduce, right? You can see that in the name, oxido or reduce. Reductase, right? So we have the oxidation reduction reaction here. It's going to help make that happen. Here we have, a lot of times we have a coenzyme. So these are things that are just helping with the enzyme. They're part of the enzyme. So they're included in this reaction, but they're going to eventually, in order for this to continue to work, right, we have to recycle this coenzyme to get this to be able to make another one of these reactions go forward. Alcohol dehydrogenase is the enzyme name, right? And here's ethanol, right? So we have this reduced ethyl alcohol compound. We have this NAD+. With this alcohol dehydrogenase, we're going to oxidize this. So then we make this oxidized product, and then we have this reduced coenzyme, and then we can use this for other things, or we can recycle it back to in this enzyme. So this is similar to oxidation reaction. So we're looking at, right, the substrate is an alcohol and it dehydrogenates it, so it's an oxidoreductase. How do we know if it's an oxidation or reduction reaction? Remember, loss of electrons is oxidation, gain of electrons is reduction. Remember, you can lose, so if oxygen is bonded to something, it pulls, right? It's more electronegative. It's going to pull electrons. We're really talking about the oxidation of carbon a lot of times, right? What's happening to carbon? Is it being oxidized or is it being reduced? When oxygen is bonded to hydrogen, right? Hydrogen and carbon have similar electronegativities, but carbon does get a little bit more of that than hydrogen. That was supposed to be an arrow that looked like that and didn't work. So we're gonna get a little more electrons with carbon. We're talking about a carbon and oxygen bond. Of course, it goes the other way around, right? We get the electrons flowing towards the hydrogen, away from carbon. So this is what we're really talking about when we're talking about gain or loss of electrons, this electronegativity difference. So whenever we have, and it's easy, whenever we have more oxygen bonds, we have oxidation. When we have less oxygen bonds, we have reduction. Do have to be careful though, because sometimes things are being oxidized or reduced because without necessarily involving oxygens. That's a good rule of thumb, but there are some exceptions and examples that you should know. FAD, FMN, for example, are coenzymes that are involved in the electron transport chain. And what we can have is basically these electrons and these double bonds can move and change where they're located on nitrogen. So nitrogen also can be involved. And we have the electrons on this nitrogen can then be when we add hydrogen bonding. So if we're going to be adding hydrogens to, that's another kind of way and I guess. So if it's not involving oxygen, also take a look at hydrogens, because sometimes we can just add hydrogens and that reduces that compound. So you might also maybe recognize that with long chains of fats, right? If we have a double bonded fat and we could actually reduce that fat, we call it reduction. If we reduce that double bond with hydrogen. So same kind of idea. So look also for hydrogens to be involved in oxidation reduction reactions as well. So if oxygen's not there, but you're adding hydrogens, a lot of times you can be pretty safe to assume that you're reducing that. So the opposite of oxidizing. Again, just kind of review this idea so you can recognize if something's oxidation reduction reaction. These things always happen at the same time. So if something is, there's something that's usually that's gaining oxygen or hydrogen bonds, and something else that's losing them. The total electrons lost by one substance will be gained by another. And then for a redox reaction to occur, something else has to accept those electrons. They can't just float around in space, right? They're lost by another substance, they're taken up by another substance. So these things are always happening together. The next class of enzyme is the transferase. So they will help to take one group from one compound and transfer it onto another. So we're going to, it's kind of like a substitution reaction, but we're basically taking, a lot of times these involve ATP. So for example, switching phosphate groups from adenosine triphosphate to a longer metabolite. And you can see that here, right? This end alcohol group on this fructose 6-phosphate sugar, we're going to transfer the phosphate group from the ATP to this sugar, this alcohol group on the sugar, and phosphorylate it. So now we're diphosphorylated fructose 1,6-bisphosphate or 2 phosphate groups. We have one on this end, one on this end. So a lot of times these are involved, and transferases a lot of times are also subclassed named as kinases. So they transfer specifically phosphate groups. The final reaction type we're going to talk about are lyases, and in this type of mechanism we're talking about an elimination reaction that occurs through mechanisms other than through hydrolysis or oxidation. So it's not through a hydrolytic cleavage or an oxidative cleavage, it's through an other type of reaction. So non-oxidative breaking of bonds. The This can also be reversible. So and typically is also going to involve the formation of double bonds and or the formation of new ring structures or cyclic structures. The term lyase is from the Greek line, meaning to break, and so that is the where this term comes from and why it's called a lyase reaction. So an example of this is isocitrate, and the enzyme that's involved in this reaction is called isocitrate lyase, and it will cleave this chemical and generate two new products, glycoxylate and succinate. You can see a new double bond is formed, and the cleavage is going to occur between these two carbons. So we're going to, the bottom part of this isocitrate is going to form the glycoxylate, and the new double bond will form between this carbon and this oxygen. And the succinate will be formed from this 4-carbon molecule, and that is how we're going to generate that new substance. So you'll have noticed through those last few slides, right? Here are the main 6 different types of classes, right? Oxidoreductase, transferase, hydrolase, lyase, isomerase, ligase. Those are the 6 groups that I talked about. But then you'll notice that, right, there were some subclass names that were put in there. Oxidases, reductases, dehydrogenases. These are doing that bigger idea kind of enzyme reaction, but they're just more specifically to exactly what's being added or done to that, right? A kinase is always adding a phosphoryl group, but right, it's transferring a phosphate group, usually from ATP. Transaminases are taking amine groups, amino functional groups, right? It's taking it from one compound to another compound, but it's still transferring a group. But we can have subclasses that give us a little more information about exactly what's being transferred. Same goes for these other subclasses. You don't need to know necessarily the subclasses. You should be able to, if I give you any type of reaction, you should be able to tell me what type of main class this fits into. You might start recognizing some of the subclasses, and that's fine. But what I want you to know for exams is that you should be able to recognize those main 6 classes, and when something's happening, fit it into one of those main 6. So for the sake of not making these too long, I'm going to stop here.